Thank you for the introduction. I'm going to talk about communication between enteric neurons and glial cells. And you've heard from the first two speakers in this session that the enteric nervous system consists of both enteric neurons and glial cells, and that they have to work together to execute all the functions of the ENS. We actually have four types of enteric glial cells that is based on their location and morphology within the plexus layers. And we know from our recent lineage tracing studies that within a single glial clone that is derived from a single ENS progenitor early during embryogenesis, that all these four different subtypes can be found, which indicates that the glial heterogeneity arises from cellular plasticity and probably local cues rather than from the niche restrictions. Today in my talk, I'm going to talk mainly about this type 1 enteric glial cell here, and it is because it is located into the myenteric and submucosal ganglia, and the cell bodies of these type 1 enteric glial cells here labeled for S100 and shown in yellow are really squeezed in between the cell bodies of the enteric neurons. And if we do some labeling for neurotransmitter release sites and we label the varicosities of neurons, we see that these really nicely decorate the cell bodies of the enteric glial cells within those ganglia. And that indicates that these cells are really perfectly located to receive signals from glial cells, and that, there is some anatomical evidence there is communication between the two cell types. I've tried to recapitulate this staining here in this ugly drawing, where there's three enteric neurons in the ganglion. There's outgoing axons, incoming axons with varicose release sites, and then there's these glial cells. And you will see this drawing throughout my presentation. Now, there's communication between enteric neurons and glial cells, and there's some evidence already from a while ago that there's, and this one is quite well established, that there is purinergic communication from neurons to glial cell, which is depolarization dependent, so we can block that with TTX. And this was actually first established in the lab of Professor Sharkey. And this is actually when there's neuronal activity, there would be neurotransmitter release, together with that would be purine release, which activates P2 receptors on enteric glial cells and giving calcium rises in enteric glial cells. Now, there's also evidence for a second way of neuronal to glial communication, and this way is TTX insensitive. And this is what we wanted to study in our current project. So for our methodology, we moved to calcium imaging, and we tried to express a genetically encoded calcium indicator, GCAM3, in glial cells specifically to be able to really look at what's happening in the glial cells and don't have any signals from neighboring neurons or other cell types. So we combined the conditional GCAM3 ROSA26 reporter line with SOX10 tamoxifen-inducible driver line, and we gave tamoxifen 2 weeks prior to experiments. You can see in this immunostaining that after a tamoxifen treatment, we reach about 96% of cells, of glial cells, to express GCAM3, but we also have about 6 to 7% of enteric neurons that express the genetically encoded calcium indicator. So for experiments, what we do is we take the small intestine of the animal, of the mouse, and then we do microdissection to isolate myenteric plexus preparations that we image under the microscope. And for the first set of experiments that I would like to show you is to really make sure that our mouse model is working, and we can do calcium imaging in this animal model. We used a local agonist perfusion using substances to activate glial cells, ATP and substance. We had also electrical stimulation of enteric neurons, so trains of electrical pulses conveyed to the interganglion connectives using the focal electrodes. So in these two movies, you can see that the system works pretty well. So you first see local activation of perfusion of ATP, and you see activity throughout the glial network, and you see that there's one neuron here expressing the GCAM molecule as well, but mostly glial cells are nicely responding. Also, if we stimulate with substance B, there's a nice and robust calcium increases throughout the glial network, and you really see, because of the confined GCAM3 expression, the nice calcium transient in the fine processes of the enteric glial cells. This gives very good amplitudes and calcium transients, and then we looked at this first way of neuron-to-glia communication. So we do this electrical stimulation of the neurons, and what you see is that shortly after or immediately during the stimulation, you see calcium increases in the remainder of the neuronal processes. So there's increases in the varicosities and also in this neuron that here, and the cell body of this neuron that expresses the GCAM molecule, but shortly after that there is a calcium rise in the neighboring enteric glial cells. And this is also evident from this movie. So you see an increase in calcium, and shortly after that there's the increase in glial cells. So this is not really new. This is what has been shown by our lab and was established in the lab of Picharki, where there's this pure energy communication from enteric neurons to glial cells. So our system works, so we can now try to focus on this second way of communication, which is TTX insensitive. But for that, we wanted to find a way or establish a way to really stimulate single enteric neurons within the ganglia of the myenteric plexus. So for that, we embarked on a calcium uncaging strategy. So the mouse system and the experimental setup is essentially the same, but now we incubate our tissues with this calcium chelating compound, which is called nitrophenyl EGTA-AM. So we incubate our tissues with that. This calcium chelator is loaded within the cells as an AM ester, and then within the cytosol of the cells it chelates calcium, but it's photosensitive. So if we hit the molecule with UV laser or near UV laser light, it's photocleaved and it releases all the calcium that it has chelated within the cell. So in that way, inducing an artificial calcium transient within the cell, and in that way we can activate neurons if you wish. So we do that using a FRAPA device on our microscope. So during the experiment, we can online draw regions of interest over the cells that we want to stimulate, and then the laser will hit these cells and photocleave the molecule. So the first thing we tried is if this was working in vitro. So we made ENS cultures. So what you see here is glial cells in culture, and this is the cell here in the middle that we uncage. You see the time-lapse images, and also here lower left. I lost my. Okay. You see the uncaging here of the cell. So we hit later that cell, and you see the calcium transient within the cell, and also spreading of the calcium increases into the processes of the enteric glial cell. You can see the traces here. If we increase laser power, we can increase the calcium transient amplitudes of the uncaged cells. So that seems to work pretty well. We also have some neurons in culture sometimes, and if we uncage neurons, then you can see this type of transient after the FRAPA glial cell gives this. There's difference in amplitude, and also, of course, the shape of the calcium transient is a bit different. We try to maximize in the rest of the experiments our uncaging efficiency by covering the whole cell body. The photo-stimulate, so the region of interest there we try to cover this maximum of the cell body. And what we do is use 75% of laser power, and we expose every pixel within this region of interest twice for 200 microseconds. So we then move to tissue preparation. So here you see an example of a myenteric ganglion where there's neurons expressing the GCAM3 molecule, and I will show you the movie where we uncage one by one these neurons that express GCAM. And you'll see increases in some of the cells, not all of them. So for example, cell 1 here that is happily spontaneously active, doesn't really get influenced by the FRAPA, which is directed at cell 1 here at this arrow. Cell 2 does not respond, but you can see it's not very happy because it has GCAM3 in the nucleus. But the other cells nicely show increases upon uncaging. So that's neurons. If we look at glial cells, so GCAM3 expressed in glial cells only, we see that again, this nicely induces calcium transients in glial cells if we direct the laser to them. We did an experiment where we uncaged the same cell twice, so to make sure that we really maximize our responses, and after the second round of uncaging, we don't see calcium transient anymore, which indicates that we really photocleave the majority of the compound at the first round of uncaging. So now, the experiment that we're really after is to look at this neuron-to-glia communication, and in this experiment, what you see here is part of an myenteric ganglion, and you see the glial cells expressing GCAM3, and these black holes, I hope it's clear from over there, that are the cell bodies of the enteric neurons. But these, of course, don't express the GCAM3 molecules, so don't see them, but they are loaded with the calcium chelator. So if we uncage a neuron that we don't see, and we look at what's happening in the glial cells, so this would be where the laser goes, we see an increase immediately after that in the enteric glial cells. So this is shown in this movie. And you see, so the laser, the white box, and shortly after that, these glial cells show a calcium transient. So what is interesting is that this calcium increase induced from this neuron-to-glia communication is really confined to the glial cells that closely unwrap the glial cell body. So what we're doing here is inducing a calcium increase in the neuron, and we see a calcium increase in this neighboring glial cell. So the first thing is that this really is confined to the neighboring glial cell. So we wanted to see if there's some neuroanatomical basis for that. So for that, we did a labeling strategy where we injected adult animals via their tail vein for a viral vector encoding for GFP, the AAV9 vector system, to sparsely label enteric neurons throughout the enteric plexus. We then did some stainings for S100 beta and really looked at how S100 beta positive enteric glial cells are touching one neuronal cell body. And we did some volumetric reconstructions, and we found that on average, there's about 2.3 glial cells touching one single enteric neuron. And this really matches well with the number of cells that we see responding after encaging. So it could be that there's some sort of neuroglia units, one neuronal cell body talking with 2 to 3 glial cells just surrounding that. Then we moved on to do some pharmacology to see what the mediators are. And this was based on, again, some earlier work from the group of Kicharki, where we could confirm that indeed it's purinergic neurotransmitters, purinergic mechanisms purines released from enteric neurons, activating purinergic receptors on enteric glial cells, and is probably releasing purines via pannexin-like hemite channels that are present on enteric neurons. So this is summarized again in this cartoon here, where you see that calcium increase would induce release of ATP via pannexin hemate channels that we have blocked with this compound called probenecid to induce calcium increases in the neighboring glial cells. So in summary, I showed you that calcium encaging can be used to induce calcium transients in single enteric neurons in glia in situ. The photostimulation of enteric neurons can activate neighboring enteric glial cells, and that this spatially confined communication might have a neuroanatomical basis in the form of these neuron-glia units that I showed you. So in conclusion, show that enteric neurons can communicate to neighboring enteric glial cells, and that this is independent of nerve conduction, and is probably by releasing purines via pannexin hemite channels. Thank you for your attention.